if you look at sequencing in 2001, this is a big genome center. There is a whole room available where you have a room of equipment where people prepare the samples, and there are about 35 people that work in here, and they prepare for 3 to 4 weeks to get the DNA into a shape that can be sequenced. And then there's another room with sequencing machines. You can see there are like 74 such big machines. It's pretty big. And then it takes about 10 people to operate this 74 machines. And for each run, you have about 15 to 40, they can run about 15 to 40 runs a day. And each instrument can produce 1 to 2 megabytes of data every day. And so this whole room every day can produce about 120 megabytes. This is already so much better than the 1970 sequencing approach, right? It was highly automated and already a lot of automation and improvement. But really the technology revolution came in 2007 when massively parallel sequencing became available. And so now for sample preparation, you need this little station and one person. They prepare the samples in less than a day. And then for the sequencing, you have this machine and it's the same guy, same technician, and he can run this, load the sample and then just go home. And the machine will run for 3 to 5 days. And per day, this machine can generate about ½ gigabyte of data per instrument. So basically they can load the sample. This is just 1 penny, and this is the sequencing flow cell. You load the DNA on one end and you will just flow through. And then you get the sequence data out directly from this machine. And so this is something I do every year. So look at sequencing last year. I do look at the updates. So this is called iSeq 100. This is like your very, individual lab can have these mini machines that runs for less than a day. It can already generate 4 million reads or 25 million reads per day. And each read is usually paired and 150 base pair long. So these are just the mini machines. And then if you have, for example, a sequencing facility, such as I think Harvard main campus have some machines, genetics department at Harvard Medical School or Dana-Farber Cancer Institute, we would have more of these, NextSeq machines, which has, you can see here, it runs for 1 to 2 days, and it can generate about 300 gigabytes of data in that 1 to 2 days, and you will get 1 billion reads from this one run, and it's paired and 150 base pair reads per run. And I just got this yesterday for the newest ANOVA Stick machine. This is really potentially for specific like sequencing companies or some big core facilities in a big university or other research institutions. So this is kind of the mega sequencing. The machine is actually not that big. It's more like a copy machine. And they can run, so per flow cell, so actually this machine can run 2 runs at a time. When you run one, you load the thing, and once it starts running, you can load another one, so you can alternate them, but you can pretty much keep the machine running all the time. And so you can see per flow cell, depending on your sequencing length, you can do short from paired and 50 base pair all the way to paired and 250 base pair. And they can also produce different amount. I guess the reagent is different. It costs slightly different, but then it will produce different amount of data. And so basically in the dual flow cell mode, so each run with 2 flow cells will generate as high as 6,000 gigabytes of data. So I remember about the paired and 150 base pair length, it's about 44 hours. So the sequencing length is proportional to how long you need to sequence this. So I guess if you are sequencing at 250 base pair, and you'll probably take maybe 60 hours. But each run will generate about 40 gigabytes of data. and at 2 times 250 base pair. And basically, in that one run, you can run 48 whole genomes and 500 whole exomes in that one run. Remember, the first human genome project, it took $30 billion over 13 years, and now it takes about $1,000, which is really amazing. And so you can look at the genome sequence cost. Yeah, so initially the human genome cost was very, very high. In 2001, with enough automation, if they were to redo the human genome project, it would only cost about 100 million. And 2007 is when the high throughput sequencing became available. You can see the price drop was huge. And this is Moore's law. Recent years, there hasn't been enough drop, but I guess because there is no competition for the market leader right now. I think with competition, they would continue to drop the price. But you can see here, we are really hovering at about $1,000 per human genome now. And so there are, again, computational effort needed, because every time you sequence one human genome is like terabytes and terabytes of data. And so there are algorithms that are needed in order to really process the data quickly. You can imagine if you have to store all the raw data, it will take a lot of space, and storage, and also processing the data takes a long time. And so in 2018, there is also an algorithm that's developed using deep neural networks to really call the mutations or germlines in the genome, because you know that between individuals in this room, our DNA sequence is 99.99 similar. And so there's no need to store all the raw data. If you only kind of capture the locations that are different between individuals, it will be a very tiny amount of data, right? So that's much better to store. And so at least this algorithm is able to use, start from the raw data and very quickly call the places where each individuals are different. And this, more and more people are looking into similar type of algorithms to call the differences between genomes. And so what are we really using these genome sequences for? The first is, you probably all knew that Angelina Jolie 6 years ago, well, now is a little over 6 years, decided to have double mastectomy because she has a female relative like mom, aunt who died of breast or ovarian cancer. She knew that she might carry risk, and after genetic testing, it was determined that she has high risk of developing breast cancer. And so she chose to have the double mastectomy to protect herself from getting cancer. And so this is, people can do this now. There are companies out there such as 23andMe. You can send your DNA from a mouth swab and you pay like $99. Sometimes they have sale, you send it over, they will sequence your DNA. And based on that, they will give you some predictions, like they tell you how much Caucasian blood or, like Asian or African blood you have, what kind of disease risk you might have. And depending on the disease risk, some people might choose to have procedures such as mastectomy or have more frequent screening to detect or prevent disease, or they might just change their lifestyle. Say some people just can't drink as much, right? Or how should they exercise and so on. So that's on the germline side. Germline meaning that the DNA that all of your cells have. But also nowadays, it's also heavily used in disease such as cancer. For example, at the Dana-Farber Cancer Institute, if a patient is diagnosed with cancer, the Farber will do the first DNA sequence for the patient for free on the tumor sample. It's not sequencing the whole genome. We usually just sequence a few hundred genes. And then based on that, to decide whether the patient should be treated with a very specific drug targeting the specific mutations. For example, if the patient have a BRAF mutation, they should be treated with a BRAF inhibitor. If they have EGFR mutation, they should be treated with an EGFR inhibitor, and so on. So you really have a more personalized drug treatment based on the mutations on the tumor. And so you can see sequencing is really changing the way how people do research, but also change how consumers or everyday people are being evaluated for disease risk or prevention or diagnosis and treatment. And so the end result is, with all this data being generated, there's a huge data challenge. And that's kind of why I hope that you should consider taking this course.